Hello everyone, welcome to Competitive Botany YouTube channel. In this video, you are going to learn competitive bits from the cell wall. To get my next video and notifications, please subscribe the channel and then tap the bell icon for notifications. Let's go to the class. Before going to the competitive bits from the cell wall, I want to give you a brief introduction regarding the cell wall. Presence of cell wall is the characteristic feature of plants. It is absent in animal cells. It provides support and rigidity to the plant body. It protects the protoplasm and prevents the movement of water into the cell. Let's go to the structure of the cell wall. The cell wall is formed during the division. It is comparatively thin in young cells than in the fully matured cells. It is differentiated into four layers such as middle lamella, primary wall, secondary wall and tertiary wall. So middle lamella, here you can observe the picture. Middle lamella is a cementing substance. The cells of the plant tissues generally remain cemented together by an intracellular matrix that is known as middle lamella. It is a viscous jelly-like glue and mainly composed of pectic compounds such as calcium and magnesium pectates. So middle lamella is a cementing substance which is present in between the two primary cell walls. Okay, so let's see the primary wall. First formed cell wall is known as primary cell wall. The primary walls of two continuous cells are present on either side of the middle lamella. That means middle lamella is present in between the two primary walls. They are composed of, that means primary wall is composed of cellulose, hemicellulose and polysaccharides, and other maybe there may be pectic substances are also present. So this is regarding the primary wall. Now coming to the secondary wall, in some cells deposit additional layers on the inner surface of the primary wall. This occurs after the growth stops or when the cells begin to differentiate. Then the secondary walls are formed. The secondary wall is usually made up of cellulose, hemicellulose and some polysaccharides. During maturity, substances like lignin, suberin, wax, tannin, calcium carbonates, they are also deposited on the secondary walls. The secondary walls, it gives mechanical strength to the plant body. The secondary wall also shows three layers such as outer layer, middle layer, inner layer. They are commonly S1, S2, S3 layers. So of these, the middle layer is usually the thickest one. Let's go to the actual content of this video, that is competitive bits from the cell wall. Let's see the first question. The structure observed by Robert Hooke under his microscope is cell wall. Robert Hooke was the first person that observed and named the cell wall. Next question. Plant cell differs from animal cell in having rigid cell wall and plastids. These two are present only in plants. They are absent in animals. Though the cell wall is rigid, but it is permeable to all substances. You should remember. The next question is, the outermost living limit of plant cells originate from. That means the cell wall is originate from the protoplasm. Next question. Plant cell walls are made up of cellulose. Animal cell is not surrounded by cell wall. Next, coming to the next question. Structural units of cell wall are cellulose microfibril. Cellulose microfibrils. Next question. Most of the abundant material found in cell wall of eukaryotes is cellulose. It is a polysaccharide. That means cellulose is a polysaccharide composed of D-glucose molecules. They are connected each other by beta 1,4 glycosidic bonds. Beta 1,4 glycosidic bonds. Next question. Primary cell walls are made up of cellulose, hemicellulose, and pectin. The primary cell wall, as I told you before, is formed first and extremely elastic, while the secondary cell wall is formed at the end of the cell growth. You should remember. They play an important role in the turgidity. Primary walls, they play an important role in the turgidity. The next question is, in an actively enlarging cell, the cell wall is grows by. The answer is intussusception. Here you have to know what is intussusception. The growth of the cell wall occurs by intussusception. Intussusception means that is deposition of cell wall material into the spaces that are present in existing cell walls is called intussusception. Next, coming to the next question. The process of intussusception is high in meristematic cells. Next question. The outermost layer of the cell wall is middle lamella. I have already told you before. The outermost layer that is present in between the two primary walls, that is called middle lamella. Middle lamellum generally occurs between the primary cell wall and the adjoining cells. Next. The first formed layer during cell division is middle lamellum. It is originated from. Answer is cell plate. Answer is cell plate. Middle lamellum is like a cementing substance. I have already told you, present between the two primary cell walls. That means in between two adjacent cells, it is a cement like substance or glue like substance. It is made up of calcium and magnesium pectates. Middle lamellum is made up of. The answer is calcium pectate and magnesium pectate. Essential mineral elements that are incorporated into the fluid plate during cytokinesis are, answer is calcium and magnesium. The next question. The precursor of middle lamellum is, answer is cell plate. Middle lamellum is formed from the cell plate. The next question. The basic unit formed for the organization of middle lamellum is, the answer is phragmoplast. Which organelle plays an important role in the formation of middle lamellum? The answer is Golgi complex. Pit membrane consists of primary walls and middle lamella. Here you have to know one thing, what are pits? Pits are the depressions or small pore-like structures formed in the cell walls due to cell wall thickenings. They are called pits. Next question is, cutinized cell walls are present in. The answer is fungi. Cutin is a modified polysaccharide and it is a polymer of N-acetyl glucosamine. It is a polymer of N-acetyl glucosamine. Let's go to the next question. Which bacterial types consist teichoic acid in their cell walls? The answer is gram-positive bacteria. Teichoic acid is a polymer of ribitol or glycerol phosphate. Plant cells that are connected by channels through their walls called plasmodesmata. Here also you have to know what are plasmodesmata. The protoplast remains connected with that of adjacent cells by delicate threads of cytoplasm. These threads of cytoplasm are called as plasmodesmata. These are called as the enlargement of cell is prevented by the secondary wall. The next question is secondary cell wall is formed between the primary wall and plasma membrane. The answer is between the primary wall and the plasma membrane. Let's go to the next question. In general, a plant cell possesses these possible number of layers in secondary cell wall. The answer is 3. Last layer of cell wall is secondary cell wall made up of 3 layers such as S1, S2, S3 layers. These are also called as outer, middle and inner layers. Next question. In dead cells, protoplast is degenerated, leaving a vacant space is known as lumen or lacuna. Lumen or lacuna. Secondary cell wall composed of, answer is pectin, soberin, lignin. The portion of middle lamellum crossing the pits become thickened, known as torus. Answer is torus. Lignified cell walls are present in. The answer is xylem tracheids. Answer is xylem tracheids. Next question. Which substance present on leaf epidermis reduces the evaporation? The answer is cutin. The answer is cutin. Evaporation of water from plant surface is prevented by, answer is cutin and soberin. Cutin and soberin. Next, cytoplasmic connections pass through. Answer is pit fields. The answer is pit fields. Cytoplasmic connections such as plasmodesmata, these are commonly called as cell bridges. These are commonly called as cell bridges.